Hello students, welcome in today's lecture. We have started our topic cell unit of life, and in today's lecture we are going to discuss the three important cell organelle, that is the lysosome, vacuoles and the mitochondria. In our previous lecture we have studied this topic up to the Golgi complex. That means in the endomembrane system, we have seen that there are four important cell organelles are involved, and those are the Golgi complex, endoplasmic reticulum, lysosomes and the vacuole. So we have already completed two cell organelle, endoplasmic reticulum and the Golgi complex. Now we will discuss the third cell organelle, which is also a part of the endomembrane system, and the name of that cell organelle is the lysosome. We have just seen some point about this lysosome. Okay, so see here, lysosome is the cell organelle. If we see the meaning of this lyso means the breakdown, or we may call as a digestion. Why? The soma means the body. Soma means the body. So this cell organelle is involved in the digestion of the material, or this cell organelle is particularly involved for the digestion of a particular subject, particular substance. Okay, so lysosome means the bodies which are responsible for digestion or the breakdown of the molecules. Okay, this lysosome was first discovered by scientist D. Dewey. The name of that scientist is D. Dewey, who first described or first discovered this lysosome. Lysosome are single membrane structure. Okay, note down here. Again I will tell you that after completing this topic cell unit of life, we have to separate, that is single membrane cell organelle, double membrane cell organelle which do not have any membrane. So here we have completed endoplasmic reticulum, which is the single membrane cell organelle, Golgi complex, which is again single membrane cell organelle, and now we are discussing the cell organelle lysosome, which is again single membrane structure. So this lysosome is the single membrane cell organelle. So very important point is that it is a cell organelle which has a single membrane, or which we also call as it is a membrane bound cell organelle, but that membrane is a single one. Okay, now this lysosome, which is a single membrane bound cell organelle, and inside this lysosome there are the different types of hydrolytic enzymes are present. Different types of hydrolytic enzymes are present. Then what is meant by hydrolytic enzyme? Hydro means water, lytic is nothing but the breakdown. So these enzymes are activated in presence of water. These enzymes are activated in presence of water. That's why we call it as a hydrolytic enzyme. We call it as a hydrolytic enzyme. Okay, so all the enzymes which are present inside this lysosome are called as a hydrolytic enzyme, and they together call as the hydrolases. They together call as the hydrolases. Okay, so hydrolases are nothing but the group of enzymes which are present inside this lysosome, which are chemically called as hydrolytic enzymes, which are mostly become active in presence of water. So they are called as the hydrolytic enzymes. Okay, now here this hydrolases enzyme which are present inside this single membrane bound structure of the lysosome, and which are those enzymes which we call as a hydrolases enzyme. So see here, the hydrolases enzymes are the lipases, then proteases, then carbohydrates. So these are the names of the enzymes. These are the names of the enzymes which we call as the hydrolysis enzyme or the hydrolytic enzymes. Okay, so these enzymes are mostly active at the acidic pH. So all these enzymes which work in these lysosomes, they are active at the acidic pH. So these enzymes work at acidic pH. These enzymes work at the acidic pH. And all these enzymes which are present in these lysosomes are capable of digesting the lipids, the carbohydrates, the proteins and the nucleic acid. Okay, so student, see here what we have seen in this the cell organelle lysosome. Lysosome means the cell organelle or the body which is responsible for digestion or breakdown of the some of the important molecules. This lysosome was first discovered by scientist D. Dewey. These lysosomes are the single membrane structure. Inside the single membrane structure, there are group of enzymes which we call as a hydrolytic enzymes, which are mostly active at the in the presence of water. They are together called as a hydrolysis. These hydrolysis enzymes include lipases, proteases, carbohydrates, and these enzymes are active at the acidic pH. Okay, so all these things are given in our NCERT book. The extra point is that the name of the scientist D. Dewey was not included in an NCERT book, but you have to remember that this cell organelle was discovered by scientist D. Dewey. Now, the point that we are going to discuss now about this lysosome is also additional point which is not given in NCERT, but you should know some of the extra points about this lysosome. That's why we are discussing here, that is the types of the lysosome. Which are the types of the lysosome that are not given in our NCERT book, but here we are going to discuss these types of the lysosome. So here there are overall about 4 types of the lysosome. But before that, one important thing that you should know, that is these lysosomes are formed from the Golgi body, or they are formed from the Golgi complex. So lysosomes are formed from the Golgi complex during the packaging of the material. Here you can get idea about why the endomembrane system includes this lysosome, ER, Golgi complex and vacuole. Here we have seen that the Golgi complex, while the packaging of the material, it formed one of the important cell organelle, and name of that cell organelle is called as the lysosomes. Okay, so here this is very important thing, that lysosomes are formed from the Golgi body or from the Golgi complex. Okay, now here we'll see why the lysosomes are called as polymorphic in nature, because if the question is asked that which cell organelle is polymorphic in nature, so here these lysosomes are called as polymorphic in nature. Poly means many and morphic means form. Poly means many and morphic means form. That means in many form this lysosome is present inside the cell. So it is called as the polymorphic. Okay, so we'll see which are the types of lysosome. That's why it is called as a polymorphic in nature. So here we'll see there are about 4 types of the lysosome. There are about 4 types of the lysosome, and those types are called as, first one is called as primary lysosome. See here, if anybody want to note down this point, because it is not given in our notes. So here the extra point that we are discussing here, that is the types of lysosome. If you want, you can note it in notebook. This the first type of the lysosome, primary lysosome. Then second type is called as autophagic vacuole. Then third type is called as heterophagic vacuole. And the fourth point is that it is the residual body. Okay, so here we will discuss in detail the types of the lysosome. But again, see here there are four types of lysosome: primary lysosome, autophagic vacuole, heterophagic vacuole, and the residual body. Sometimes this autophagic vacuole and the heterophagic vacuoles are included in the type secondary lysosome, and this residual body is called as the tertiary lysosome. So see here, this is the primary lysosome, it is called as the secondary lysosome, or we may say secondary, and this is the tertiary. Don't get confused, okay? You may call that there are three types of lysosome: primary lysosome, secondary lysosome, and the tertiary lysosome. Primary lysosome, name itself given is a primary lysosome. In the secondary lysosome, there are two types of lysosome: autophagic vacuole and the heterophagic vacuole. And in the tertiary lysosome, there is a type another that is called as a residual body. Or you may can remember that lysosomes are of the four types: primary lysosome, autophagic vacuole, heterophagic vacuole, and the residual body. Okay, now students, here I will explain these types in detail, that is what is meant by primary, auto, hetero, and the residual body. Okay, so see here, this we will discuss this first type, that is called as primary lysosome. See here, this is the sac which is called as a single membrane bound structure, and in that sac there are hydrolytic enzyme which we together call as a hydrolysis enzyme, but these enzymes are in inactive form. These enzymes are in inactive form. Okay, they are not active, or they are not able for digestion of a particular substance which comes in contact with it. ओके, त्याच्या कॉन्टॅक्ट मध्ये कुठलाही सबस्टन्स आला तर त्याचं डायजेशन करण्यासाठी तो सक्षम नाही आहे. इट इज नॉट एबल, अँड दॅट्स वाय दोज एन्झाइम्स विच आर प्रेझेंट इन दिस लायसोसोम्स आर कॉल्ड ॲज द इनॅक्टिव्ह form of the enzyme. So all enzymes which are present in the lysosomes are called as an inactive form of the lysosome. Okay, so here you say that this primary lysosome means the lysosome in which the enzymes present which are called as a hydrolysis enzyme which are in inactive form are called as the primary lysosome. Okay, very clear. We have to remember it in only one sentence that lysosome which has the inactive form of enzyme is called as the primary lysosome. Now the second point is that the autophagic vacuole. Then what is meant by autophagic vacuole? Again here, this is the lysosome which is full of the enzyme. Okay, it is present in a cell because it is one of the important cell organelle in the eukaryotic cell that we are going to discuss here. Okay, so any eukaryotic cell, maybe plant cell or in the animal cell, in the cytoplasm there is a cell organelle which we call as a lysosome, and in that cell organelle, if any cell organelle, maybe endoplasmic reticulum, chloroplast, mitochondria, Golgi complex, any cell organelle even become old one. Okay, so old cell organelle we have to remove from that cell, because if we remove that old cell, then and then only new cell organelle will be formed at that place. Okay, म्हणजे एखादी सेल ऑर्गॅनेली कुठलीही आपण म्हणूया एंडोप्लाझमिक रेटिक्युलम, गोल्जी कॉम्प्लेक्स किंवा मायटोकॉन्ड्रिया, क्लोरोप्लास्ट किंवा स्वतः लायसोसोम असेल. ओके, ह्या सेल ऑर्गॅनेली ओल्ड झाल्या की त्या डिस्ट्रॉय करायला लागतात, त्या सेलमधून बाजूला टाकायला लागतात आणि त्याच्या जागी नवीन त्या सेल तयार व्हाव्या लागतात, नवीन सेल ऑर्गॅनेल तयार व्हावे लागते. म्हणजे जुनी ओल्ड ER जर बाजूला केली तर त्या ठिकाणी नवीन न्यू ER cell organelle तयार होऊ शकते. So to remove that old cell organelle, the lysosome secrete their enzymes on that particular cell organelle and it digests that cell organelle. So here the lysosome which contain its own cell organelle, such a lysosome which contain its own cell organelle, that means it digests its own cell organelle. Okay, The lysosome which digests its own cell organelles. ज्या सेलमध्ये लायसोझोम आहे त्या सेलमधल्या सेल ऑर्गॅनेलिज त्याने एंगल्फ केल्या, इंजेस्ट केल्या आणि त्याचं डायजेस्ट केल्या, तर त्या लायसोझोमला आपण काय म्हणणार आहोत? वी कॉल इट ॲज अ ॲटोफेजिक व्हॅक्यूल. We call it as a heterophagic vacuole, which is the second type of lysosome. Then what is meant by heterophagic? Atto means self and phagic means to eat. Okay, when lysosome eat its self cell organelle, then that lysosome is called as heterophagic vacuole. And see here we have given the name here to this lysosome, that is called as heterophagic vacuole. Student, one more thing you have to remember here, that due to this ability to eat its own cell organelle, lysosomes are also called as the suicidal bags. Lysosomes are also called as suicidal bag. कारण लायसोझोम जेव्हा त्याच्या सेलमध्ये एखादी सेल ऑर्गॅनेली डायजेस्ट करतो तेव्हा त्या सेल ऑर्गॅनेली बरोबर ते होल सेल डायजेस्ट होतं, ते होल त्या सेल बरोबर ते स्वतः लायसोझोम डायजेस्ट होतं. अँड दॅट्स वाय लायसोझोम्स आर कॉल्ड ॲज दी सुसायडल बॅग्स. ड्युरिंग द डायजेशन ऑफ द अनदर सेल ऑर्गॅनेली इट इज सेल्फ डायजेस्ट विथ दॅट सेल ऑर्गॅनेली, सो इट इज कॉल्ड ॲज दी सुसायडल बॅग. ओके, सो दिस इज द सेकंड टाइप ऑफ lysosome, that is called as the heterophagic vacuole. Now the third type of lysosome, that is called as the heterophagic vacuole. Then what is mean by heterophagic vacuole? So see here, this is again the lysosome which is a single membrane structure, and with this lysosome if any foreign body, with this lysosome if any foreign body means what? Any foreign organism like bacteria, viruses, fungi come. कुठलाही फॉरेन ऑर्गॅनिझम जर या लायसोझोमच्या जवळ आला तर लायसोझोम काय करणार आहे? इमिडिएटली एंगल्फ दॅट फॉरेन मटेरियल इनसाइड इट अँड डायजेस्ट दॅट फॉरेन मटेरियल विथ इट्स एन्झाइम. ओके, so this property of the lysosome, when it comes in contact with the foreign organism like the pathogenic microorganisms which cause a particular disease in particular animal, so these foreign organism which may be bacteria, viruses, fungi, protozoans, this foreign material when comes in contact with this lysosome, they engulf that foreign material, and such a lysosome which engulf the foreign particles, mainly the pathogens, then such a lysosomes are called as the heterophagic vacuole. So this is called as heterophagic. Then what is the meaning of heterophagic? Hetero means different and phagic means again to eat. Hetero means different and phagic means to eat. So it eat different particles other than its own cell. Okay, so these are the particles which are foreign to this lysosome. They are not its own cell particle, but here it eats own cell organelle. That's why it is called as heterophagic, and it is different from their cell, so it is called as the heterophagic vacuole. Okay, you can clearly understand this concept. Then fourth point of the lysosome is called as the residual body. It is called as the residual body. Then what is mean by the residual body? So see here, this is the lysosome. This lysosome contain the different material in it, and the material which is not digested by the lysosome, and we may say that the undigested material which is present in the lysosomes are called as the residual body. See here, this is the heterophagic vacuole. Here its own cell organelle are digested, but the undigested part of this cell organelle come in the cell. Here this is a heterophagic vacuole. Here also foreign particles get digested, but undigested foreign particles comes in this lysosome, and so the lysosome which contain the undigested food material, undigested substances are called as the residual body, and it is the fourth type of the lysosome. Okay, so students, here out of the 90 questions which are asked for the NEET exam, if one or two questions are asked from out of the NCERT syllabus, then if any question is asked on this lysosome about this content, then you have to remember that lysosomes are the polymorphic in nature. It is found in different form because these lysosomes are of the 4 types, are found in the 4 form. Primary lysosome, which is the lysosome where the enzymes present in it are in inactive form. Heterophagic vacuole are the lysosome which digests its own cell organelle. Heterophagic vacuoles are the lysosomes which contain the foreign particle like bacteria, virus, fungi, etc. and digest it. And the residual body are the lysosomes which contain undigested food material inside it. So here we have completed why the lysosomes are polymorphic in nature, because it is found in 4 different forms. Okay, and what is the function of this lysosome? Lysosome's function is digestion. Whatever it may be which comes in contact with lysosome, it gets immediately digested. It may be food, it may be foreign organism, or it may be any other structure which is digested by this lysosome. So here we have completed the lysosome part, which is one of the important cell organelle, belongs to the endomembrane system, and this lysosomes has a various type of enzyme, or which we may call it as a sac which is full of the enzymes. Okay, now after the lysosome, the another cell organelle is vacuole, which is also a part of the endomembrane system. So next to lysosome, the cell organelle is vacuole. So students, can you recall what is vacuole? Because we have drawn one diagram of the animal cell, and for homework I have given one diagram of plant cell that you have to draw for yourself. Okay, that's our question. But here vacuole means what? If we compare the plant cell and the animal cell, if we compare the diagram of the plant cell and animal cell, you can observe that in a plant cell, suppose this is the diagram. Okay, so you have to see only the concept of the vacuole, not we have to focus on the diagram of the plant cell. But see here, if we consider it is a cell of the plant, then in the plant almost 90% volume of the cytoplasm is occupied by this vacuole, and that vacuole is called as a central vacuole. So major part of the plant cell get occupied by the portion or by the cell organelle, and the name of that cell organelle is called as the vacuole. Okay, so what is mean by vacuole? Vacuole is not, again, it is a single membrane structure. It is single membrane structure. Vacuole is a single membrane structure. These vacuoles are found both in plant and animal cell. They are present both in plant and animal cell, because for neat point of view, if the question is asked that where the vacuoles are found, vacuoles can be found both in plants and animal cell, but the number is more in the plant, or size of the vacuole is larger in the plant as compared to the animal, because mostly animal have very few vacuoles, or if vacuoles are present in the animal, they are very small in size. But in the plant, vacuoles are more in number and their size is also large, and it is mostly the volume of the cytoplasm is occupied by the vacuole. So vacuole are present in the cytoplasm of the cell. Again, they are single membrane structure. Now if you see the chemical composition of this vacuole, then what you will observe that inside this vacuole there will be present the water, sap. You will found the excretory products of that cells are stored in that vacuole. So next is excretory products. And the waste matter which is not required to that cell is also get stored inside this vacuole. That means this vacuole are the cell organelle which stores water. Excess water is always required to the plant, and that excess water in the plant is stored in the vacuole. So see here student, one of the reason is why vacuoles are larger in size, why vacuoles are more in number in the plant, because plant require large quantity of water as compared to the animal cell. Okay, so large water is there in the plant, that's why vacuoles are also large, and mainly in the vacuoles water gets stored, so there are more vacuoles in the plant as compared to the animal. कारण animal एवढी वॉटरची गरज लागत नाही जेवढी प्लांटला त्याच्या ग्रोथ आणि डेव्हलपमेंटसाठी लागत असते, कारण प्लांट वॉटर रूट मधून ऍब्सॉर्ब करतात and it gets stored inside the vacuole. Okay, so here water is present in the vacuole. Sap, what is meant by the sap? Sap means when the minerals or salts get dissolved in the water. When minerals get dissolved in the water, then it is called as the sap. So sap means water plus minerals. Excretory products of that particular cell, which is a waste matter which is harmful to that cell, is called as the excretory products. And the waste matter which is not required for particular cell is get stored inside this vacuole. Okay, so it is also important that which content of the cell is present in the vacuole. So for need point of view, which point is important? It is a single membrane structure and it is found in both plant as well as animal, but more in plant as compared to the animal. Water, sap, excretory product and waste matter is present in the vacuole. Now the membrane which we call as a single membrane, and the single membrane of the vacuole is called as tonoplast, is called as the tonoplast. So it is very also important for need point of view. What is the name of the single membrane which is present around the vacuole? It is the tonoplast. And the function of this tonoplast is that this single membrane which we call as the tonoplast present around the vacuole is facilitate the transport. It is responsible for the transport. Which transport is carried out by this tonoplast? So this tonoplast is important for the transport of ions, is important for transport of ions and other molecules. Vacuoles are responsible, tonoplast is important for transport of ions and other molecules against the concentration gradient. Against concentration gradient means in the function of the plasma membrane we have seen that along concentration gradient means from higher concentration to the lower concentration, but against concentration gradient means from lower concentration to the higher concentration. So this tonoplast is responsible for the transport of ions or other molecules. Okay, but how this transport takes place? Against concentration gradient, that means from the lower concentration to higher concentration, or we may write here from lower concentration to higher concentration. Okay, it is called as the against concentration gradient. So if we see the concentration which is present inside the vacuole, it is always higher, because all the ions, all the particles which always move from lower concentration to higher concentration. So all these ions from low to high concentration move inside this vacuole through this tonoplast, and that's why the concentration of that particular ion substance is higher in the vacuole as compared to the outside surrounding environment, because this tonoplast is responsible for movement that is lower to higher in the vacuole. Okay, so this is also important that tonoplast is the name of the membrane around the vacuole, and this tonoplast is important for transport of ion or other molecule from lower concentration to the higher concentration. So all the concentration of that ions are present inside the vacuole, and ultimately that concentration is higher inside the vacuole. Okay, and here we have completed the part vacuole, and what we have seen in this vacuole. So this vacuole is the important storage organelle. We store water, we store sap, we store auxiliary product, we store the waste matter. Large amount of vacuoles are found in the plant. About 90% volume of the plant cell get occupied by this vacuole. But see here, one more thing we have to remember inside the vacuole, that where this vacuoles in animal is present. So here if we see the example of the animal, that means can you remember the protista that we have seen in the biological classification. So see here students, amoeba, which is a protozoan, okay, example from the kingdom protista. So in this amoeba you can observe contractile vacuole. In this amoeba there is a contractile vacuole, and which is involved for the excretion. Okay, so excretion is a mechanism where in that amoeba there is a vacuoles are formed, and that vacuole contains excretory material which is given outside of that cell by the process of exocytosis. Removal of the material outside the cell, which called as a through the membrane, particularly through the tonoplast, we call it as a exocytosis. Or in simple word, you can remember that in the amoeba the vacuole present, which is called as a contractile vacuole, which is responsible for excretion of the waste matter. While in some protista, then do not get confused, amoeba and protista are the same examples of the kingdom. In protista you can observe the food vacuoles. You can observe the food vacuoles. When the protista engulf food, then in that protista you can observe in the cytoplasm there are the food vacuoles. That means in that vacuole the food of that protista get stored. Okay, so this is about the vacuole, which is the important cell organelle present in the cytoplasm. Here we have completed the 4 cell organelle, which is the part of endomembrane system: ER, Golgi complex, vacuole, and the 4th one is the lysosome. Okay, now the next cell organelle, which is not a part of the endomembrane system, it is a separate cell organelle, but it is very important cell organelle because it is the energy, or it is the power of our body, of our cell, and the name of that cell organelle is the mitochondria. So see here, mitochondria. 2 more things that are not given in our textbook, NCERT book. You have to remember that this mitochondria was first discovered by scientist Colliker. This mitochondria was first discovered by scientist Colliker. But the name mitochondria was given by the scientist Benda. Okay, so see here, who discovered this mitochondria first? Scientist Colliker, and the name mitochondria was given to it by the Benda. These 2 names are not given in our book. Another important thing is that without staining lysosome, we cannot observe it under the microscope. So to observe this mitochondria, we have to use the microscope. And here you should also know which stains are used to stain the mitochondria, and then only we can observe this mitochondria under the microscope. And the name of that stain is Janus green B. So all these 3 points are additional point that you have to remember. If you want, you can note it. So Janus green B is the name of the stain which is used for staining of the lysosome, and it is generally blue or purple in color, and we can easily observe the microscope by staining with this stain. See here, the number of this mitochondria is variable in different cell because of their physiological, or it depends on the physiological activity of that cell. Number of the mitochondria is variable in different cell. It is not constant in all the cell. प्रत्येक सेल मध्ये मायटोकॉन्ड्रियाचा नंबर कॉन्स्टंट पाहायला मिळणार नाही, तुम्हाला तो व्हेरिएबल पाहायला मिळणार आहे. का व्हेरिएबल पाहायला मिळणार आहे? कारण जशी जशी त्या सेलची फिजिओलॉजिकल ऍक्टिव्हिटी आहे, त्या सेलचं मेकॅनिझम आहे, त्या सेलचं फंक्शन आहे, त्याप्रमाणे आपल्याला लायसोसोम त्या सेलमध्ये पाहायला मिळणार आहे. नॉट ओनली नंबर बट इट्स शेप, इट्स साईज इज ऑल्सो व्हेरिएबल. त्याचा शेप आणि त्याची साईज सुद्धा तुम्हाला काय पाहायला मिळते प्रत्येक सेलमध्ये. इट इज व्हेरिएबल. व्हाय? बिकॉज अगेन द फिजिओलॉजिकल ऍक्टिव्हिटी ऑफ द cell defines how many mitochondria will be there, how much or how its form or how its shape it will be there. That all will be determined by the physiological activity of that cell. So number, size and shape of the mitochondria is variable. Okay, generally if we see this mitochondria, then it may be found in the sausage shape, or they are in the form of cylindrical shape. सॉसेज म्हणजे बशी म्हणतो आपण, चहा पिण्यासाठी जे आपण वापरतो, दॅट इज कॉल्ड ॲज अ सॉसेज. ते तुम्हाला बशीच्या फॉर्ममध्ये मायटोकॉन्ड्रिया पाहायला मिळतील किंवा सिलेंड्रिकल स्ट्रक्चर तुम्हाला सेलमध्ये मायटोकॉन्ड्रियाचं पाहायला मिळतं. ठीक आहे, तर आता आपण मायटोकॉन्ड्रियाची डायग्राम काढत त्याचं एक्सप्रेशन बघणार आहोत. इथे पण तुम्हाला कळलं, कॉलिकरनं पहिल्यांदा डिस्क्राईब केलं, पहिल्यांदा शोधून काढलं, बेंड्यानं मायटोकॉन्ड्रिया नाव जे आहे ते दिलं, and Janus green bee stain was used for staining of that particular mitochondria. This mitochondria has generally sausage or the cylindrical shape, and this mitochondria's number, their shape, their size is variable. It depends on the physiological activity of that particular cell. Okay, see here. If we see the structure of mitochondria, see carefully, if we see the structure of mitochondria, then what you will observe? Here, this is the mitochondria. Okay, I will draw the rough sketch of it. Can you observe it? It is a shape of this mitochondria, and here you can observe such a finger-like projections inside this mitochondria. Okay, so these finger-like projections are called as the cristae. So students, see here. This one is called as a matrix of mitochondria. Inside the matrix you can observe single circular DNA. You can observe the ribosomes. These ribosomes are of the 70S type ribosomes. Here you can observe this is outer membrane. This one is inner membrane. Inner membrane shows finger-like projection. Those finger-like projections are called as cristae. Here, this portion is called as matrix of the mitochondria. Some RNA molecules can be also observed inside the mitochondria. So here this mitochondria has a double membrane structure. Only one cell organelle that we are going to first, which has the two membrane, outer membrane and the inner membrane, and that is the mitochondria. So mitochondria is a double membrane structure, and previous cell organelle that we have studied, those are in the single membrane structure. So here if we see the outer membrane of this mitochondria, then it is get continuous one. It is not interrupted. It is circular in outline. It is continuous. But the inner membrane of the mitochondria, shows such a finger-like projection, and those finger-like projections are called as the cristae. Okay. So if the question is asked that which membrane of the mitochondria shows finger-like projection or cristae, then the answer to it is the inner membrane. Now the fluid which is filled inside this inner membrane, and that fluid is called as the matrix. That fluid is called as a matrix, and if we see the composition of the matrix, then you will find that in the matrix there is a single circular DNA molecule is there. There is a type of ribosome that is called as 70S type of ribosomes are present. There are some RNA molecules are present. There are some protein molecules are present inside this matrix. Okay. So here one important thing that you will be noticed that DNA, mitochondria has its own DNA. In previous cell organelle, in not any cell organelle we have seen that there is presence of DNA, but here the DNA is present inside the mitochondria. So if the question is asked that whether the DNA is present in the mitochondria, so answer is yes, because it contains its own DNA which is in the form of circular DNA. Okay. Now here the important thing that we have to discuss is that this mitochondria is called as the powerhouse of the cell. It is called as the powerhouse of the cell because it is a main cell organelle which is involved in aerobic respiration. Then what is meant by aerobic respiration? Respiration is intake of O2 and giving out of CO2. Respiration process is of two types, aerobic and the anaerobic. When oxygen is required for respiration, it is called as aerobic respiration, and when oxygen is not required, it is called as anaerobic respiration. So with the help of oxygen, when respiration process is carried out, that respiration is called as the aerobic respiration. And in presence of aerobic respiration, this mitochondria produce ATP, and that's why it is called as powerhouse of the cell. So important thing is that why mitochondria is called as powerhouse of the cell, because this mitochondria produce ATP inside the cell. It produce ATP. Okay. Mitochondria produce ATP, that's why it is called as powerhouse of the cell. Why it is called as powerhouse of cell then the production of ATP? Because ATP are the energy coins. When your body or whenever the cell require energy, that energy is fulfilled by this ATP. So our energy source is ATP. Without energy we cannot carry out our single activity, and for that the ATP are required, and this ATP, which is the power of our body, is produced by this cell organelle, and that's why mitochondria is called as the powerhouse of the cell. Okay. Why it is called as powerhouse of the cell? Because the main power of our body is our energy, and that energy is provided to us in the form of ATP, adenosine triphosphate, which get produced inside this mitochondria. This outer and the inner membrane has its own enzyme, and all those enzymes are required for carrying different activity of this mitochondria. So outer and inner membrane, both these membranes are full of enzymes, and all these enzymes are required in carrying out the function of this mitochondria. So see here you can see that this double membrane structure of the mitochondria divide the lumen of mitochondria into two compartments. Inner mitochondria divides this mitochondria into inner compartment, and here this outer mitochondria or outer membrane divides the mitochondria into the outer compartment. See here you can see the space in between outer and inner membrane, and that space is called as intermembrane space. That space is called as intermembrane space. Okay. So which points are important from mitochondria? Double membrane structure. The space between outer and inner membrane is called as intermembrane space. Mitochondria's inner membrane is in the form of cristae, which are nothing but the finger-like projection. Inside the mitochondria, matrix is present, which is the fluid part of the mitochondria. In the matrix, circular DNA is present. In the matrix, you can observe 70 type of ribosome, protein molecules, RNA molecules. While this mitochondria produce the ATP, which is a part of our aerobic respiration, or mitochondria produce the ATP, which is the energy coins. That's why it is called as powerhouse of the cell. And mitochondria is a cell organelle important for aerobic respiration. That means the respiration which is carried out with the help of oxygen, that is called as aerobic respiration. So students, see here in today's lecture we have seen the lysosome, vacuole and mitochondria in detail. So here we will stop today's lecture, and here we completed the mitochondria part of the cell unit of life. Thank you.